Hi friends, in the previous video we have learnt about the structure of a neuron, but it's most important to learn how the nerve impulse, that is the electrical or the saltatory impulse, and the chemical impulse gets transmitted across a neuron. The neurons actually are arranged in the form of cells in a battery, one after the other, where we can take the cyton to be the positive end and the axon end to be the negative end. So the axon of one neuron would be lying very close to the cyton of the next neuron, and so on. So in this video, I'm telling you a little bit about chemical synapses that actually take place at the axon ending. Usually, the ends of the axon are dilated to form a knob-like structure that is called a synaptic knob. So this area that you see dilated over here is actually the synaptic knob. In this, you can see that there are various vesicles that are rounded structures. These are termed as synaptic vesicles. Inside these, there are as many as 10,000 molecules of chemicals that are termed as neurotransmitters. Now, these are very interesting molecules because, as the name suggests, a neurotransmitter is something that facilitates the transmission of the impulse across a neuron. If you observe this particular diagram carefully, you can make out that this membrane is called the presynaptic membrane. This actually is the cell membrane of the axon ending of one neuron. And lying close to it is the postsynaptic membrane, which actually is the cell membrane of the dendrite of the next neuron. So this basically is referring to a junction between two neurons, wherein this part is the axon of a neuron, and this part would actually be the cyton of the next neuron. The question now comes how the impulse gets transmitted across this axonic area to this particular area. Since I talked to you about these vesicles which are containing substances like neurotransmitters, for example, acetylcholine, which happens to be an excitatory neurotransmitter. On the other hand, we even have inhibitory neurotransmitters, for example, dopamine. So what happens is that as soon as the impulse comes across the ending of the axon, that is the synaptic knob, there are some calcium channels which are located over here, something similar to the sodium and potassium channels that we have in the neuron. So these calcium channels open and a lot of calcium rushes inside from the extracellular fluid inside. This calcium that rushes inside the synaptic knob actually facilitates the synaptic vesicles to bind with this particular area of the cell membrane. This area that you can see between these two is referred to as the synaptic cleft. Cleft usually refers to a space. Over here, the cleft refers to this synaptic area which is lying between the vesicle and the postsynaptic membrane, that is the next neuron. So as soon as the calcium channels come inside, these vesicles, they bind with this cell membrane and a lot of neurotransmitter gets poured into the synaptic cleft. But just by accumulating here, the problem does not solve because the impulse needs to cross this and enter into the next neuron. Now friends, if somebody comes to your house, you actually need to recognize that person, only then you can let that person in. Same way, if there is a substance lying here that wants to trigger some sort of a potential into the next neuron, then first of all this substance must be recognized by something on the cell membrane. And usually we have a huge array of proteins lying on the cell membrane, and these specialized proteins that can actually recognize this neurotransmitter substance are known as receptor. So receptor is basically anything that will actually recognize a protein, the neurotransmitter substance. So as soon as this gets poured in, the receptor, it recognizes it, the acetylcholine, dopamine or any other neurotransmitter which has been poured here will bind to the receptor and then trigger the electrical potential in the next neuron by opening the sodium gates or the voltage gated sodium channels, and this is how the impulse would get transmitted to the next neuron. Whatever I have talked to you about, friend, is summarized over here in this diagram. Depolarization across a neuron opens the calcium channels towards the axonic terminals or the synaptic knob, due to which lots of calcium from the extracellular fluid diffuses inside. This leads to the fusion of the synaptic vesicle with the presynaptic membrane, and then the neurotransmitter which is contained inside the synaptic vesicle binds with the receptors on the next neuron or the postsynaptic membrane. That opens a lot of sodium channels, and then the action potential actually goes across this. Thanks for watching this video, friends, and stay tuned for watching the next videos in which we are going to discuss in detail the saltatory conduction and other aspects of the nerve impulse transmission. Bye-bye.